logistics setup. Okay. Oh, I have to find my mouse because I have to accept. There. I cannot see my mouse for one or the other reason. Okay, this is really annoying. Somehow with the presenting, I lost my mouse. So okay, there it is. I had it for a short bit. There. Yes. No. Yes. Okay.
Okay, we can see our first slide.
Yeah. So okay, let's get started. So the main topic of the research is in our lab is perception, active perception, and for this we use the whiskers of mice. And why is that? Well, let's start with a human example. So if you find yourself in a room with the light switched off, I cannot really say dark room because in the Netherlands that has a very different meaning that I won't discuss here. So you enter a room with the light switched off and you want to find the light switch, right? So imagine what you would do. Well, typically what you would do is you make very large movements with your hands. Well, in order for yourself, not to walk into something, but also to find a wall, because normally that's where light switches are. And then once you find a wall, you make kind of smaller movements with your hands to feel whether you can find a light switch. And once you find it, you make even smaller movements with your fingers to find out, how it works, what kind of light switch it is, and then, well, you push the button. So what this tells you is basically two things. First of all, is that we need sensory perception, and we make a model of the world to kind of make a model of where the walls are, where the light switches are, what kind of room it is, and so on. But also that this sensory perception is a very active way of it's a very active process. So of course the feelings that the sensations that we get through our fingers, through our arms, depend on how we move our hands, but we move our hands and arms depending on where we think a wall might be or where the light switch might be. So there's a continuous feedback loop going on between what we think is there in the world and how we position ourselves, how we position, in the case of vision, our head, our eyes, and so on. So there's this constant feedback loop. It's not like we just sit there and wait for sensations to find us. So we need this active sensing to optimize our perception. Now, funny thing is that animals, this is a rat, but mice also do the same thing. So this is a video from the Prescott lab. You see, this is actually, I pause it, this is actually at 1/7 the speed, because normally the speed at which rodents use their whiskers is 50 hertz. So it's way too fast to see. But what you see is that if it explores this kind of cup, it makes very small movements with its whiskers. But when it goes out into this big open field, it makes very, very large movements with its whiskers, as you can see here. So it's the same as what we do with the large movement with our arms and the small movements, for the walls and the light switch. The rodents do the same thing. Now, so that's one of the reasons why it's so good to study this behavior in rodents. And here you see a mouse in a gap crossing task. So it's dark, but this is infrared light. And there's a gap here, and the mouse has to find the platform with its whiskers. And then when it knows where it is, then it jumps. So you see here that actually it's a good sensory modality to observe, because you can really track these whiskers in the dark, which is a natural situation for mice. They live, naturally in holes below the ground. And the funny thing is, even though whiskers are obviously specific to rodents, to mice and rats and stuff, the way the cortex is organized for the whiskers is actually pretty similar to how our sensory motor system for our fingers and our hands. And of course, with mice, if we study these kind of systems in mice, it's a very natural sense, and we have all these kind of tools that we can use, like optogenetics and Krelog's mechanisms and so on, that we wouldn't be able to use, for instance, in humans. And again, it's a very active sense. So we already in our lab, it was discovered that mice control the whisker position to where they think an object is. I'm not going to go too much into details for it now, but you can play them little tricks, and you can see that over a couple of whisking cycles, they really target their whiskers to where they think an object is. Now, a very quick introduction to the whisker system. So what you see already also in the videos, when a whisker touches an object, the curvature of the whisker changes. Now, the whisker, it's a hair, a stiff hair, and it's inside a follicle, it's inside the skin, and around surrounding the whisker, where the whisker is inside the skin, there's these mechanoreceptors, basically these press sensors. So once the curvature of a whisker changes, then the whisker starts pressing against these mechanoreceptors, and that is then the sensation that enters the nervous system. Now, the whiskers are organized in this grid-like manner on the snout of a mouse. And the funny thing is that this grid-like structure is conserved throughout the brain for mice. So each whisker has its own little designated brain area in the brainstem where the signal goes first. They're called barrelets. You can see that in picture number B. Then it goes to the thalamus, where it also has this topographic organization, where it's called barreloids, what you see in C. And then it goes to a part of cortex, the somatosensory cortex, where they're called barrels, which we see in picture D. So it's kind of easy to target the correct area for the brain, because it conserves this kind of this topographic structure. So you can really see in the brain which part of the brain belongs to what whisker. Now, what do we do in my lab? Of course, if you want to study, how this works, how this somatosensation works, you can do this on all kinds of levels, from all the way from molecules all the way to behavior, which is completely on the right-hand side. I don't really seem to have a pointer, so I'll just give visual clues. So you can study it on the level of synapses, of neurons, of networks. And if you really want to understand it, of course, you would have to study it at all levels, but that's hard to do. So we try to kind of push from different angles and see what's going on. So we'll quickly, in this talk, I will focus a bit on neurons, networks, and also on behavior a little bit. But we also use molecular tools in the department to study this behavior. So if we look at what we do in my lab, is we start with information methods, like analysis methods, and we make single neurons models of that, and then we go with increasing abstractions from more biologically realistic network models to more coding, more abstract models, all the way to try to understand behavior. And in this talk today, I would, because half an hour is limited in time, so I will focus a bit on the single neuron data analysis part. So if you look at neurons in the cortex, most of you will know that you have inhibitory neurons and excitatory neurons. And these obviously differ in the effect that they have on other neurons. Interneurons have a negative effect on other neurons, and excitatory neurons have a positive effect on other neurons, but they also differ in other ways. So these excitatory neurons, or principal cells, as we call them, they connect mostly between different networks in the brain. So from layer to layer, or from thalamus to cortex, whereas interneurons connect more locally within a network. Also, interneurons connect basically to all neurons in their surroundings, whereas principal cells, excitatory neurons, tend to connect to more specifically to neurons with certain properties that respond to certain types of input. And these excitatory neurons, they respond to more specific stimuli, so they're more tuned, as we say in neuroscience, whereas inhibitory neurons respond more randomly to more different types of stimuli. So that's what we know about kind of the network properties of these neurons, but we also know that they differ in their intrinsic properties. So the question that we started out with is, how can we measure how the information transfer, what kind of information they transfer, what kind of information they pick out, how much information they transfer, and how their intrinsic properties, rather than the network properties, influence the information transfer. So to do that, we needed obviously to measure information transfer in these neurons. So basically, you give them an input and you see how they spike, and then you say, okay, how much information about the input is there in the output spike frame? But the problem is as follows, and we had to develop a new method for that. So basically, if you more generally, if we take a step back, you can say, okay, in neuroscience, we typically talk about encoding and decoding. So basically, encoding means, okay, that there's an input coming into the brain or into a neuron, and then, you know, that the brain does something with that, and then you record some neural activity. Now, how is that neural activity generated? That is what we typically call encoding. You can also ask the opposite question. So, okay, given that I record the neural activity, can I kind of deduce what the input was? And that's what you typically call decoding. So what does the neural activity tell me about the stimulus? And of course, the brain does both all the time. So it generates all this neural activity, and it sends all these signals to next processing layers. But it also has each processing layer basically has the task of kind of getting back to, okay, what does it mean that the input that I receive, what does it mean, and what information do I need to get out and transfer to the next level. But here we talk about information coding, we talk mostly about the decoding part. So basically, what we assume in neuroscience is that there's some sort of relation between what's happening in the outside world, how I move my arms, the things that I feel with my fingers, light that hits my eyes, and the neural activity that my cortex displays. And you call the relation between this, you call that neural coding. And you can basically look at two things. You can look at what information is coded. So basically in neuroscience, we talked then about receptor fields, like what is it that neurons respond to? What is the information they extract? And you can also ask how much information is encoded and how much information is lost. Maybe the system discards a lot of information. And that's also an interesting question, like how much information does it actually need to keep? Now, and if we talk about information, there's a very precise mathematical definition of information. I mean, we of course talk a lot about information in a very loose sense. It's the information age, the information that's in my phone, but mathematically or physically, it actually has a very specific meaning that you can describe mathematically. If you say, okay, if I have all the possible inputs that a system can receive, and all the possible output that a system then can generate, and then you can say, okay, if there is a relation between the two. I hope that there's a relation between the two. If there's no relation, then apparently the output has no information about the input. But if there is some relation between the two, then exactly that is like, how much do I know about the input if I observe the output, or how much do I know about the output if I observe the input? That is what we call information. So basically, information is just an advanced way of a correlation. So I really like this image from Wikipedia because it shows you the difference between a correlation and information. So basically, in information, what we measure is if there's a relation between input and output. But that correlation assumes that relation is linear, is like a line. And in mutual information, we can say, okay, this relation can be anything. So here in the bottom, you see all kinds of relations where the information would be a value, which you say, okay, this is a system that has information about the input, but a correlation would give you nothing, just as an example. So how do we measure that in vitro experiments? Basically, the question is, okay, what is it in the biophysics of a cell that influences information processing? What is the information loss of the spike generating process? And then the problem is that if you do in vitro experiments, that cells only stay alive for about 1 hour. It depends a bit on what kind of stimuli you give them. And then you want to test multiple settings. So maybe you want to, add dopamine or serotonin or whatever to your system, and then see how that influences information transfer. And you want to give the cell a stimulus that's kind of realistic, that looks a little bit like what it would normally see. Now, there have been several ways, there have been proposed in the past, there have been several ways to do that. So here you see a very nice, very beautiful experiment by Rob de Ruiter van Stevenink and Bill Bialek in the 80s, where they had a blowfly and they recorded from a single cell in the visual system of this fly. And they showed it a random pattern that changed in velocity. So here you see in this graph, you see on the horizontal axis, you see the time in seconds. On the bottom, you see the spikes of the neuron, and then you see in black, you see the velocity of the pattern system, and then they fitted a model to this, and then they reconstructed the input. So in light gray, you see the reconstructed input. So and that way you can say, okay, I give the input, then I estimate how much I can reconstruct the input from the spikes, and then that can give me an estimate of information. You can see here already that there's also kind of a relation between information and signal to noise ratio, so that you can use that sometimes. So that works very well, but you need it to do this. You need to fit a model. In this case, they used spike triggered average, and the fitting of this model needs a lot of data. So you need to do this 1 hour of recordings to have enough spikes to fit this model. Well, the problem is by that time, your neuron is dead. So you have one estimate of a mutual information, but you cannot say, okay, how does it change if I do something? Because by that time, your neuron is not alive anymore. There's another method called the direct method that was also by the same Rob de Ruiter van Stevenink, where they said, okay, we attack this problem differently. Is we give a little bit of stimulus, but repeat a short stimulus. So it was the same thing: the blowfly, the single neuron, the moving random dots. But we repeat the same stimulus over and over again. So here you see the stimulus, and then in the bottom you can see for different repetitions of the trial when the neuron spiked. You see that there is a good deal of regularity in it, but there's also some deviations from trial to trial. And then you can, again, from basically the signal to noise ratio, you can estimate how well you can deduce this input from the stimulus. But again, you need about 1 hour of recording. So we have still the same problem. Now, so this is the setting where we started, and the problem is that classical methods need about 1 hour for a single information estimate. And we cannot do that because then our cell is dead. So what we did is, and I'm not gonna completely go into all the details of this, but what we did is, instead of trying to estimate the information between this random stimulus that you see here kind of in the middle and the spike train, is we said, okay, we do something else. We let this random stimulus be generated by an artificial network, and this artificial network responds to a very simple stimulus, and that's a binary stimulus. It's just randomly put to 0 or 1, and it flips randomly between this 0 and 1 states, and then there's some artificial neurons that respond to that then generate this artificial stimulus. So instead of having to find the relation, which is very complicated, and we need a lot of data for that, between this complicated stimulus and the spike train, we can now find the relation between the simple stimulus, the up-down stimulus, and the spike train. And we can find the relation between the simple stimulus and the complicated stimulus. So we have now two information estimates: the amount of information in the input and the amount of information in the output. And then by comparing the two, we see how much information is lost. So by doing this little trick, by relating it to this binary signal, we can now all of a sudden control the information in the input to begin with, by the switching speed of this binary state, and by how many neurons and how many spikes there are in this artificial network. But we can now estimate the information in about 5 minutes instead of 1 hour. So now we can all of a sudden start recording and doing all these experiments where we add dopamine, or we change something about the experimental setup, and we see how this influences the information transfer. And we have an optimal response model for that called the Bayesian neurons. But that's so if you want to try this method, you can. It's actually all the code is there on GitHub. if you would like to try that. So we did that. We recorded about 300 neurons with this method. We have about 850 trials for this. And we share all this data. So if you want to play around with it, go ahead. It's all online. And this is kind of how it looks. So inhibitory neurons, they fire a lot more. They fire a lot faster. So that was also a good thing about this method, because we could, for the inhibitory neurons, use a faster switching stimulus than for the excitatory neurons, because they in barrel cortex, excitatory neurons, they fire at 2-3 hertz. You can do whatever you want to them, they don't fire faster. So here you see the stimulus. On top you see the slow one for the excitatory neurons. On the right you see the fast one for the inhibitory neurons. In gray you see it. In gray and white you see the switching of this binary state to on and off. And then the bottom you see the response of these neurons. So you see that they respond indeed on average when the hidden state is on. And now we can indeed estimate the information transfer in these excitatory and inhibitory neurons. Inhibitory neurons are here blue, excitatory neurons are red. And what you see is that these inhibitory neurons, surprisingly enough for me, transfer more information. So their spike trains contain more information, even though they are not the ones connecting to the next level. So the excitatory neurons are much more selective in what kind of information they send than the inhibitory neurons. What you see also is that the excitatory neurons, they have a much higher threshold, which is kind of expected because those are the ones that fire much lower. So we have learned now a little bit about the difference in excitatory and inhibitory neurons, because I did not really go into the variability, but you saw also that the responses of the excitatory neurons is much more variable. So what you saw is that the excitatory neurons are actually more variable and more selective. So they really select a specific message to send to the next level. Whereas these inhibitory neurons that collect mostly locally, they basically update or keep track of all information that's in the network, which then makes you wonder a little bit about their role. Maybe they have a gating role where they have to, keep track of everything that's going on and only allow certain excitatory neurons to fire. Now, if we now add, for instance, so we did a lot of experiments with dopamine. So if we add a dopamine receptor agonist, then you see that the inhibitory neurons, as you can see here, they increase both their firing rates and their information transfer. So you see here in the leftmost figure, you see on the bottom you see the increase in firing rates, and on the vertical axis you see the increase in information transfer. So you see that in blue, the inhibitory neurons, with dopamine, they increase their firing rate and they increase their information transfer. Whereas excitatory neurons, they decrease their firing rate and information transfer. Although they're so spread out, it's kind of hard to tell. Then you also see that they increase the inhibitory threshold, as you can see on the rightmost figure. So you also see that the dopamine the activation of the D1 receptor has also a very different effect on inhibitory neurons than in excitatory neurons, because in inhibitory neurons, they increase their firing rates and they lower their thresholds. And we saw, actually, I'm not showing the pictures here, but we saw the same for the D2 agonist. So basically, I'm almost finished, not to worry. We also approach the same from the completely other side, from the behavioral part. So I showed you already before this kind of gap crossing task. So we saw that if an animal is on a platform, it has to find the other platform. It's in the dark with its whiskers, and funnily enough, they're motivated enough. Maybe they find the experiment boring or something. That when they know where the target platform is, they jump. And again, this data is shared if you want to play around with it. And what we see, if we locally inject dopamine or D1 agonist into these animals while they do this task, they become, you could say, more daring. So they need less touches, less touch duration, they wait shorter, whatever way you measure it, they jump faster. So we see now that with this method, we can approach basically a problem from two sides. On the behavioral level, we see that animals jump faster, so they integrate information faster for one reason or the other. Whereas dopamine on the single cell level, influences mostly inhibitory neurons and make them fire faster and transfer more information. So how does this work? How does changing these single cell properties result in somehow more daring animals? Well, if you want to answer that question, we have to go to the network level. And that is actually what we're doing right now. So we're trying to build networks to kind of explain, artificial networks to explain this. How can a single neuron property trend, change in properties of a single neuron, how can it result in a change in behavior? So these are the three PhD students there. I saw that at least some of them are also here today. They're working on this problem. So basically, Tusif is looking at networks from a plasticity point of view. So you can also, and he's trying to find kind of patterns in these networks. So you can also cut away. So it's I mean, if I would cut off your finger, that would be an extremely painful thing to do. But for a mouse, if you cut off their whisker, it doesn't hurt them and they grow back. So you can do that for a while. So you can see how does the brain reorganize itself once you cut off one of these whiskers. And he's looking at the patterns in the cortex when that happens. Nishant is really trying to fit models of the things that I just showed to you, of the single cells, and can we understand what is happening in these cells that makes them fire with a lower threshold, with transfer more information and so on? Can we really understand what's going on in these cells? And then Nicolas is really trying to build a biologically realistic. So we have a model already of the barrel cortex, but we don't have a model yet of the thalamus and of the brainstem. So he's really trying with the data that is also available from the LM Brain Atlas to understand, to build the models of what's going on the lower levels. Yeah, and we're trying to, on a network level, understand what is going on here. So actually, I wanted to stop here. There's more I can tell, but maybe I would here just give you the opportunity to ask you for you to ask some questions. So yeah, I mean, because I was asked to keep it at only half an hour, I thought it would be good to just stop here. So thank you for listening, and yeah, go ahead.
Thank you, Fleur. Go ahead with the questions, guys, and if you